Well, good morning, everybody. It's a real pleasure to be here. I very much enjoyed the day yesterday and today as well. I'm going to tell you a little bit about our line of thinking that we currently have based on the experience that we have with both large-scale Eqtl analysis and single-cell Eqtl analyses. So I suppose this is something where quite a few of us would want to get. Take a GWAS, take the associated variants and try to identify the causal genes inside the loci and get also insight into the downstream consequences, hopefully to arrive at a limited number of genes that really drive disease. And one strategy that we took is to do a large-scale Eqtl meta-analysis across 37 blood-based cohorts, in total close to 32,000 samples. And what we did cis-Eqtl analysis, trans-Eqtl analysis. We also studied 1,200 traits, calculating polygenic risk scores for each of these 32,000 individuals and ascertaining whether polygenic risk is correlating with gene expression levels. And when having a sample size like this, it doesn't come as a surprise that you find cis-Eqtls for nearly every gene. But still, if you are thinking about trans-Eqtls, it's only a minority of the genes for which we can now detect trans-Eqtls. We studied 10,000 variants from the GWAS catalog. It was a release 2 years old, ascertaining which of those are giving trans-Eqtl effects, observing that 36% of those are affecting gene expression levels in trans. But what is clear, as you can see, is that trans-Eqtls are very difficult to detect in blood if those genes are hardly expressed. And I think it's extremely valuable to also consider strategies in other tissues where large sample sizes are necessary, but probably achievable in order to identify a lot of trans-Eqtls of the other cell types that are of interest. A few examples. I show a few examples here on cancer. Various risk factors associated, for instance, to multiple myeloma, affecting the same gene in trans. The same story for Hodgkin's lymphoma, also various variants on different chromosomes affecting the same genes in trans. And sometimes also unexpected effects, for instance, a testicular germ cell tumor risk factor giving effects in trans on a few genes that are specifically expressed in testis, showing very low expression in blood, but still then enabling us to identify the trans-Eqtl effects of this testicular germ cell tumor risk factor. The same story for melanoma. One thing that we observe quite often is that many different genetic risk factors converge on the same genes. So multiple independent variants on different chromosomes affecting exactly the same genes in trans. And that was one of the reasons for embarking on polygenic risk score calculations in correlation with gene expression levels. So if you do this for instance, systemic lupus erythematosus, you see that there are various loci affecting predominantly interferon genes. And if you calculate a polygenic risk score in these 32,000 individuals, ascertain which genes they are correlated with, you also see that increased genetic risk for SLE is giving increased expression of interferon genes. So as a proof of concept, whether this strategy might also help you to identify genes that really drive specific phenotypes, we have one example on various plasma metabolite levels. We calculated PRS scores based on GWAS studies that have been conducted on 4 metabolites, and what we observed was that these polygenic risk scores significantly affect the expression levels of 2 genes, and those 2 genes happen to be essential enzymes in the formation of these different metabolites, indicating that indeed this strategy is able to inform yourself about genes that could play a causal role in different phenotypes. So this is about what we've recently done. What we observe is that cis-Eqtls are ubiquitous. We find them for a lot of genes. That in contrast to trans-Eqtls that are less easy to detect. The effect sizes are usually small and they are highly cell type specific. But if you have a sufficiently large sample size, it's possible to actually start finding them. And I think they are extremely relevant for better understanding disease. But there are a few challenges that we encountered. We had 4 different technological expression platforms, Illumina, 2FE arrays, and RNA-seq data that we had to integrate. That did take a lot of work, but eventually it turned out to be a feasible strategy. And another problem that we ran into, and I suppose that's going to hold for other future functional genomics strategies aiming at bringing a lot of data together, is that we had to go for a federated approach because it was impossible to bring all the raw data together. And this is something where various avenues exist to ease also these types of federated functional genomic analyses. And a preprint is available, and all summary statistics data, including all non-significant results, can be downloaded from the Eqtl Gen website. And I do think that there are now a few opportunities based on these kinds of approaches. And one of them is to identify regulatory networks. And that's based on various recent observations. One thing that we observed is that effects on gene expression levels are also highly context-specific, not only cell type-specific, but also highly context-specific. One example is shown here for a lipid variant affecting fatty acid desaturase expression levels. And you can see that that cis-Eqtl effect is strongly influenced by the expression levels of another gene, and that's a sterol binding transcription factor. So it's the combination of both. And that sterol binding transcription factor actually is binding on top of this SNP that's affecting the FATS2 gene. So the combination of them, the levels of the transcription factor in conjunction with the particular lipid SNP, that combination is subsequently determining what is the activity of the FATS2 gene. So if you think about it in a slightly different way, you've got, through this approach, the ability not only to identify downstream effects of individual variants, but you also have now a strategy to identify the upstream regulators, while still having this SNP that permits you to anchor it and also to make inferences on which allele is increasing and which allele is decreasing expression. So if you think about that, let's suppose you've got a gene for which you've, through this approach, identified an upstream gene, but now let's suppose this gene itself, this downstream gene, is an upstream regulator of another gene. So then you can start thinking through an approach like this and can keep on going, and through this approach you will be able to start making causal regulatory functional inferences. So one thing and one dream that we have is, would it be possible to start integrating that? And I was very fond of the word that Jesse mentioned, the inference engine, and I think that is definitely something that will be very welcome. Linking SNPs through various functional genomic approaches, for instance Eqtls to individual genes, using networks to infer what are the implicated pathways, but then also including this directionality information for better identifying what are the really crucial genes that drive disease. And one thing that is going to help this tremendously, I believe, is to go with a lot of single cell expression data. So one study that we recently published was only on 45 different individuals on PBMCs, and we were able to identify cell type specific cis-Eqtls. But I think the most interesting thing was the fact that we observed that different individuals showed completely different co-expression networks. So one example is shown here. We've got a type 1 diabetes SNP that's strongly affecting RPS26. If you break it down into individual samples, for instance, the individuals that have the TT genotype, you see that that relationship between RPS26 and another gene, in this case RPL32, is strongly positive, whereas the individuals that have the CC genotype show a negative relationship, indicating that this type 1 diabetes is not only affecting RPS26 and cis, but it's also having an effect on the regulatory wiring. It's changing the relationship between various genes. And that supposes that it could well be that we also are, in this room having different regulatory networks. And through single cell Eqtl or expression analysis, it's possible to start identifying those differences in the regulatory wiring as well. But the thing is, we did this in only 45 different individuals, enabling us to identify this relationship. Well, we could replicate this finding in bulk RNA-seq data, but then we needed a lot of extra samples. So there's clear merit in going for the single cell route when it comes to doing Eqtl analyses. And I think, and this is really provocative, and I don't think it's going to fly soon, but you can imagine that if you were to have two different individuals with two different regulatory networks, could it be that if you look into John, where we've got a drug that's targeting this green gene that ultimately is signaling through this key driver gene, that for John, this drug is helping to cure him, whereas due to the absence of this relationship in Kate, this drug actually is not going to work. Once you were to have these networks in a lot of different cell types and tissues, that you could start tinkering whether this might actually hold for certain drugs. So one thing that we've recently initiated is a single cell Eqtl Gen Consortium. to perform large-scale single-cell Eqtl meta-analysis in PBMCs, to build these personalized regulatory networks, identify these co-expression QTLs, and subsequently, based on the insight that we gained there, apply these models to individual genomes to predict individual cell type regulatory effects and gene expression levels. And I think that ties in to some extent with the ambition that is being outlined here today and yesterday of the Common Disease Consortium, because how my dream, let's keep it for myself, is, I would think it would be really fantastic if we would be able to start initiating generation of large-scale population-based single-cell multiomics data in a lot of different individual cell types. And the material for that, it can be primary cells, it can be organs on a chip, as Siska indicated. There are clear merits to that, but it's something like this, a bold initiative that might, help us get further. And I think, that would ultimately permit us to generate a lot of personalized cell type regulatory networks, and that could also be of help to aid in drug discovery and better tailoring of drugs to different individuals. So that is what I wanted to say to you. Thank you very much.
Thanks, Lude. Any questions for Lude? Manos?
So Lude, I think that as a sort of an ideal scenario, I think it would be great to do single-cell Eqtls or single-cell analysis in as many tissues and individuals as possible. But practically, do you believe that the technology is actually ready to do that in terms of actually representing the full transcriptome or other molecules that we want in some of the single-cell perspective? Because from our experience, the number of genes that you access, even if you just sum up all the cells, 3, 4, 5,000 cells in a single, you get maybe 3,000, 4,000 genes that you can properly quantify, even if you sum up all the reads. So could we do this and be comprehensive, keeping in mind, of course, that we don't even access splicing, right? So
sure, yeah. Well, so, either the glass is half full or half empty. So I think, you're absolutely right. There are quite a few challenges right now. But you also need to keep in mind is how quickly the single cell field has progressed in the last couple of years. So if you would sort of, project how it's going to go, let's say in 2 or 3 years, I think it's pretty realistic to assume that, in 2 or 3 years, we will be able to quantify in a single cell, let's say 10,000 genes at once. But it's either you are optimistic or a little bit more pessimistic on that. That's
Well, I'm optimistic. I'm talking about the immediate future.
Yeah. So if you want to engage in this today, no.